Hi, my name is Robert Malone. I'm a physician scientist. I'm the original discoverer of mRNA vaccines, mRNA as a drug, DNA vaccines, and that whole suite of technologies. I was asked by Guillaume to speak to you at VivaTech, and I'm glad to do so, regarding his first question being, could I explain how messenger mRNA and that suite of applications were first discovered? It goes back to my time as a graduate student between 1987 and 1989 at the Salk Institute, working in the laboratory of Dr. Inder Verma in the molecular biology and virology laboratories of the Salk. I was asking questions concerning how mRNA is packaged to form retroviral vectors and developing and testing technologies for large-scale synthesis of purified RNA and delivery of that into cells so I could ask these fundamental questions about structure function. I was working closely with a postdoc in the lab, Dr. Dan St. Louis, who had been investigating the use of retroviral vectors to transfer genes into cells and then transplant them into mice so that they would continue expressing the transgene. Dan had run into a problem. The transgene expression in the mice was lost after about 3 weeks. The dominant explanation was gene silencing. It was my insight that the timing fit exactly with what one would expect for an immune response against the transgene, and that turned out to be the problem. From that, it was a short step forward for me as somebody focused on gene therapy, working in this leading gene therapy lab, to recognize that gene transfer and all of the associated technologies could be used for generating vaccine responses. I subsequently had discoveries relating to the use of lipofectin for delivery of mRNA into cells, and then tadpoles, frog embryos, and chick embryos. And from that, it was a short step to the original disclosures of the use of mRNA as a drug, and then subsequently the use of mRNA for vaccines. This was further developed when I left the Salk and joined a tiny startup called Vical, which filed together with the Salk all of the original patents that form the basis of the patent estate that Merck and Vical used for 20 years to try to develop DNA and RNA vaccines and related products. So the short version is it was a series of findings, including at Vical, where we had the discoveries of naked DNA and naked RNA delivery that formed the founding intellectual property for this whole suite of technologies.